Hi, welcome back to LPHumananatomika.
The components of cell. Each cell consists of cell membrane, nucleus and cytoplasm. Going in detail one by one, we will start with cell membrane. The cell membrane is the outer limit and is semi-permeable. It is also called as plasma membrane. The electron microscopy of a cell reveals the principal components of plasma membrane, which are lipids, proteins and carbohydrates. The lipids include glycerophospholipids and cholesterol. The proteins include integral proteins or transmembrane proteins and peripheral proteins. The carbohydrates include glycolipids and glycoproteins. The proportions of protein, lipid and carbohydrate in the plasma membrane vary with cell type. But for a typical human cell, protein accounts for about 50% of the composition by mass. All types of lipids account for about 40%, and the carbohydrates comprise the remaining 10%. However, proteins and lipid concentration varies with different cell membranes. For example, myelin, an outgrowth of specialized cell membrane that insulates the peripheral nerves axons, contains only 18% of protein and 76% lipid. The mitochondrial inner membrane contains 76% protein and only 24% lipid. The plasma membrane of human red blood cells has 30% lipids. Let us see the structure of lipids. The cholesterol molecules are less abundant than glycerophospholipids, accounting for about 20% of all the lipids. It helps regulate the stiffness of membranes, while other less prominent lipids play roles in cell signaling and cell recognition. Glycerophospholipids are the most abundant lipids in cell membranes, accounting to 80% compared to cholesterol. They are insoluble in water, but they are having unique geometry makes them aggregate into bilayers without any energy input. This is because they are two-phased molecules. When we look at the structure of a single glycerophospholipid molecule, it is composed of two major regions: a hydrophilic head, which is water loving, and hydrophobic tails, which are water fearing. The hydrophilic head is composed of a choline and a phosphate. This head is connected to a glycerol with two hydrophobic tails called fatty acids. The whole structure is called as the glycerophospholipid. Hence, the hydrophilic heads of the glycerophospholipids in a cell membrane face both the water-based cytoplasm and the exterior of the cell, and their tails, hydrophobic carbon tails of fatty acids, line up in the bilayers interior, which does not have any interaction with water. Hence, phospholipids go on to form a great lipid bilayer cell membrane, separating fluid inside the cell from the fluid outside the cell. The second main component of the cell membrane are proteins, which account for roughly half the mass of most cellular membranes. Few of the proteins traverses only a portion of the membrane linking with one layer, whereas others span from one to another side of the membrane, thereby exposing to both sides of membrane. Proteins that are associated with the membrane but are not inserted into the lipid bilayer are called peripheral membrane proteins. They are usually bound to other proteins in the membrane. Some of the protein molecules of the inner layer of plasma membrane provide anchorage to the microfilaments and microtubules, to form a filamentous network which act as cytoskeleton to alter the cell shape or impart movement of the cell and provides attachment sites for transmembrane proteins. The proteins embedded into the membrane and sticking out on both sides are called transmembrane proteins or integral proteins. The portions of these proteins that are adjacent to hydrocarbon tails have hydrophobic characteristics, and the parts that stick out of cell membrane are hydrophilic in nature. Numerous transmembrane proteins act as channels permitting selective permeability to ions like sodium, potassium, calcium, chloride and bicarbonate. The passage of smaller molecules like glucose, amino acids and nucleic acid precursors also take place through these routes. Each channel is specific for one variety of ions or molecules. Some channels are continuously open called leak channels, whereas others may open and close called gated channels. If the gated channels open due to alteration of membrane voltage, it is called voltage gated channel, or if they open after binding to chemicals, it is called as ligand gated channel. Some channels open when the membrane is stretched. Some of the transmembrane proteins act as carriers because they transport materials across the plasma membrane by binding ions and other molecules and change their configuration for transport. Molecules move from areas of high concentration to areas of low concentration down the chemical gradients. Cations move to negatively charged areas, whereas anions move to positively charged areas down their electrical gradients. Transport occurs in two different ways, passive and active. In passive transport, molecules are transported from high concentration to low concentration region. For this action, no energy is required. Two types of passive transport are observed. Transport to which no protein is needed is called diffusion. When carrier proteins move substances down their chemical or electrical gradients with the help of transmembrane protein in which no energy input is required, it is called facilitated diffusion. Glucose transport down its concentration gradient from extracellular fluid to the cytoplasm of the cells is a typical example of facilitated diffusion. In active transport, the molecules move from low concentration to high concentration region. For this, where the carriers transport substances against their chemical and electrical gradients, it requires energy which needs input of ATP to produce high energy. This type of carrier proteins are called pumps. One of the classic examples is sodium potassium activated adenosine triphosphate, which is also known as sodium potassium pumps. A carrier protein acts in three different ways: unipodes, sympads and antipodes. Unipodes transport only one substance down the concentration gradients. Glucose transporters act as unipode. Sympads carry two substances at same time in same direction down the concentration gradient. Example for this is sodium and glucose, which are transported together from the intestinal lumen into mucosal cells. This is also an example of facilitated diffusion. Antipodes allow the movements of two substances in opposite directions. The activity of sodium potassium at ATPase is classical example of antipode. It moves sodium out of the cell in exchange for each potassium that moves into the cell. Another way of the cell membrane which transports molecules inside and outside is through the process of endocytosis and exocytosis. Endocytosis means uptake of substances or materials which can be solid or liquid from the exterior to the interior of the cell. During this process of endocytosis, the cell membrane locally invaginates or infolds, forms a depression and surrounds the particle that is engulfing. It then pinches off to form a small sphere of membrane called endocytic vesicle that contains the molecule and transports it to wherever it will be used in the cell. Intake of solids like microorganisms by this method is known as phagocytosis, or it is called as cell eating, and of fluid is known as pinocytosis or cell drinking. The endocytic vesicles are respectively called phagosome and pinocytic vesicle. Cells can get rid of or throw waste products or molecules through exocytosis, which is the opposite of endocytosis. Exocytosis is a process of release of the contents via membrane-bound secretory vesicles from within the cell to the exterior. During exocytosis, vesicles come to the surface of the cell membrane and merge with it. The cell membrane then invaginates, pinches off and release their contents to the outside of the cell. The third component of the plasma membrane are carbohydrates. They are generally found on the outer layer of the cell membrane and linked either to lipids to form glycolipids and or proteins to form glycoproteins. Carbohydrates like peripheral proteins lead to the formation of concentrated sites on the surface of the cell which identify each other. This identification is crucial to cells as they permit the immune system to distinguish between the own cells of the body and the foreign cells or the tissues. Both glycolipids and glycoproteins are together known as glycocalyx, forming the cell coat, which is extremely hydrophilic in nature, attracting huge quantities of water on the cell surface. This helps the cell to interact with its fluid-like environment and also in the ability of the cell to acquire substances dissolved in water. Many tissue antigens, including major histocompatibility antigens, are located in the cell coat. The plasma membrane and the glycocalyx complex exert an electrostatic force to bind the identical cells to form specific tissues and helps in active transport of sodium and potassium ions across the membrane. Recent evidence suggests and proposes the fluid mosaic model of plasma membrane in which proteins, phospholipids and cholesterols are constantly moving. This movement helps the cell membrane maintain its role as a barrier between the inside and outside of the cell environments. The cellular membrane relies on natural biophysical properties to form and function. At physiological temperatures, cell membrane are fluid. At cooler temperatures, they become gel-like. The functions of the cell membrane are: it maintains the shape of the cell by anchoring to the cytoskeleton. It associates with other cells and extracellular matrix through intercellular junctions to form tissues. It provides microenvironment or fixed environment for cell function. It protects the cytosol of the cell from external environment. It forms a stable barrier between two aqueous compartments which are towards the outside and inside of a cell. The membrane also assists the proteins to monitor and maintain the chemical climate of the cell with the shifting of molecules across the membrane. The embedded proteins perform specialized functions which include cell-cell recognition and communication, which helps tissues work together as one. It contains receptors and enzymes which help in cell signaling and cell metabolism, which leads to alteration of DNA synthesis, gene expression, protein synthesis and calcium regulating process. It regulates the selective transport of molecules. Cell membranes serve as barriers and gatekeepers. They are semi-permeable, which means that some molecules can diffuse across the lipid bilayer but others cannot. Small hydrophobic molecules and gases like oxygen and carbon dioxide cross membranes rapidly. Small polar molecules such as water and ethanol can also pass through membranes, but they do so more slowly. On the other hand, cell membranes restrict diffusion of highly charged molecules such as ions and large molecules such as sugars and amino acids. The passage of these molecules relies on specific transport proteins embedded in the membrane.
If you like the video, hit the like button. If you have not yet subscribed, subscribe to the channel and hit notification button. Join my private Facebook community where like-minded students will support, discuss, encourage and push each other towards reaching the goal. For link, visit the description area. Thank you for watching.